Okay, take a seat that we start with the last session of the day.
So welcome to the last session of the day that is entitled AI Application Part 2. I am Marco Stellaro from the Department of Information Engineering in Padova, and co-chairing it with me is Paolo Ravazzani from the CNR in Milano. So,
welcome to this session. And I think we can start to the session, and so we can start with first invited talk with the first invited speaker, Professor Riccardo Bellazzi. The platform is yours. Professor.
Thank you.
Professor Riccardo Bellazzi is Professor of Bioengineering and Biomedical Informatics and Director of the Department of Electrical Computer and Biomedical Engineering at the University of Pavia. And this contribution will be focused on AI in action, deploying trustworthy AI medical systems with reliable components.
Thank you for the invitation, and it was really nice to put together the program together with the other organizers, and I'm really happy to be here today. So you can see my presentation as a sort of natural follow-up of the last presentation, so it's a really continuum in terms of the topic that we are dealing with. So I would like to focus my presentation on the problem of using day by day the AI medical systems and talking a little bit about reliability, just to put another term on the table. So the start is basically to take into account that even if we probably don't have a real groundbreaking application in the clinics of AI, it is true that the number of medical devices that have been approved by FDA is constantly increasing, and this is the same also for European Union. Of course, now with the AI Act, we have learned it yesterday, probably there will be, different patterns to get CE markings, but still, there are a huge number of products that are now ready to be sold and available in the market. If we take a look at the last snapshot of FDA in terms of AI and machine learning enabled medical devices, we can see that we have more than 500 products in the field of radiology. Radiology and the image analysis seems to be the most important area for the AI at the moment, and this is the result of the breakthrough that occurred in the area of image processing and using convolutional neural networks and other models to analyze images. We have also another area that is cardiovascular diseases, and it's probably related also to signal processing and other areas that are slowly arriving. But what does this mean? Actually, it means that we have a number of products that are used day by day in the clinic. And therefore, we have to move to the scenario that was presented by the in the previous talk. That is, we typically, as machine learner, put our attention on validating our models and validating on the bench, but we have to move them on the bed. And actually, yesterday, there was a question about validation. Probably we haven't talked a lot about validation of machine learning models, but there are now standards for performing this type of validation, but most of them are related to the issue of validating the machine learning model before it is sold, before it is part of the product that goes into the market. So my talk today is related to the last part of the validation, that is what is called the online evaluation of our model. Online evaluation means that we have a model that we are using, and we need to validate the model while we are using it. And there are several reasons why this is very important, and are common issues that have been mentioned also in the last talk. So first of all, we want to monitor the performances of the model because we want to avoid a number of problems that may occur, and probably they are going to occur frequently. So first is poor results transferability, and this is true because we change, let's say, the environment from the environment for which these tools have been developed. There is a famous failure of the EPIC model for sepsis prediction that has been published recently that showed that the model that was part of the solution that was sold by EPIC, which is one of the most important vendors all around the world of electronic health record, turned out to fail when it's used in real practice. And this is exactly a problem of poor model transferability. Then we have another issue that I'm going to talk about, that is samples out of distribution. It is related to the single case that we have to analyze and for which we have to perform the prediction. And that may turn out to be out of distribution of the statistical distribution that was used to learn the model. Then we have, along the time, potentially population and data drifts that are related to the fact that changes, that the situation changes over time, and also the population that comes to my hospital changes, and the environmental situation changes, and that we need to monitor this. And finally, even our concept, so our target class, may turn out to be different over time. So the definition of the disease in which we are interested in may be slightly different over time. So we definitely need to monitor the performance of our models. And then we have another issue that is related to the overall validity of the model. And we need to decide what we want to do in order to make alive, in a way, our model. And there are two general strategies. One is to try to implement a solution for continual learning or adaptivity of our model, so learning while working. The other strategy is, after a while, retrain the entire model or say, okay, the product doesn't work anymore. So let's move on to the first topic, and I'm not going to talk all of them. I just want to do a little deeper dive on the issue of samples out of distribution. So in my group, we believe that the problem of detecting the samples that are out of distribution, the patients that are out of distribution, are part of the trustworthy components that we would really need to implement an AI system in which we trust. We had a very interesting discussion yesterday afternoon about trustworthiness and how trustworthiness can be affected by the new AI Act in a way. So if we stay with the seven principles for trustworthy artificial intelligence, there is one that is technical, and many others are socio-technical, and this is perfectly right that AI, to be trustworthy, need to have a socio-technical system that is trustworthy, made of people and tools. Since I'm an engineer, we are, of course, interested in trying to improve what we can in terms of technological tools. And we are working on the technical robustness and safety component of trustworthy system. So if we read the points related to technical robustness and safety, we find the resilience to attack and security, the general safety of the tool, of course, the performance, the accuracy, but also there is the term reliability and reproducibility. And we think that the reliability concept itself is a subtle concept that is related to a property that our AI system should have in order to be able to detect situation in which my model doesn't work for a specific patient. So we know that machine learning models typically, after having been trained, give a probability, for example, a probability of the disease given the data that are collected on a specific patient. The problem is, if the probability is 0.9, we can say, okay, fine, we have a model that predicts with a high certainty that the patient has a certain disease. But how we can ensure that the 0.9 has been correctly estimated? So it's the right number, it's not the wrong number. And it might be the wrong number because our patient is different from the patient that has been used to train the model, or because there are unfairness, hidden unfairness reasons behind. We had a very nice discussion about unfairness yesterday. So our idea was to try to detect if it is possible to find those examples that are unreliable on a point-wise manner. And we define reliability relying on two different principles. On the one hand, the so-called density principle, that is the new instance close to the training distribution, or it is out of distribution? In the plenary this afternoon, we have heard about the problem of out-of-distribution samples. The other aspect is related to the fact that if the feature space is very high, is huge, then we may have locally properties of our classifier that are such that the samples are typically not properly, let's say the probability related to the class of the sample is not properly estimated in that subspace. So what we would like to have is to be able to detect these two situations. So how to do it? There are different strategies. What we wanted to do is to basically devise a potential strategy that we may ask as an additional property of the classifiers of the AI systems that are put into the market. And in particular, the idea is that they should be able to provide also instruments to assess the reliability of the prediction. And in particular, if we think about the density principle, a potential solution that we have tested in Pavia is to rely on generative models. So this is a sort of straightforward idea because our idea is to train an autoencoder on the training set, which of course will be never given to the, let's say, together with the tool in general. But this autoencoder represents a model, basically, of the data and of the data distribution. And thanks to the autoencoder, it is possible to define a mean square error of the prediction, so how the data belonging to the training set can be reproduced by the autoencoder themselves. Then, given a new instance to be predicted, what we can do is basically to try to understand if this data, if this new instance will be properly predicted, so the attributes will be probably predicted by our autoencoder, will be correctly predicted with a low mean square error. If this happens, then of course the example is reliable and we can perform our prediction. Otherwise, the prediction turns out to be unreliable, and it's a matter of understanding what to do with these unreliable predictions. So if we want to see in action this kind of principle, we have performed a test on a database that was collected by the Brain Teaser project. You will hear more about Brain Teaser in the next presentation, next two presentations, second presentation after mine. Some data have been collected and made available for machine learning challenges, and one of the problems was to perform prediction of the situation of the patient, and in particular of the worsening of the condition of the patient using the expanded disability status scale. And the idea was to basically build a classifier in order to predict if the patient will be going to cross the threshold at least twice of the DSS, that is the disability scale, within 1 year. And actually looking at the predictions after detection of reliability clearly shows that reliability is a tool that is able to detect the patients that are well described by the trained model, while if we consider situations in which there have been significant drifts, then on average the performance of the model are very weak. So having available this kind of tool, in our idea, should be one of the preconditions in order to be able to, let's say, put into the market a tool and give more trust into the prediction themselves. The next point that I want to touch with the presentation is related to the problem of retraining the model. So the idea basically is to understand when it's necessary to retrain the model, or if it's better to perform a continual learning. And as many of us experienced, pandemic was not only bad, but it was also little good for testing ideas. And we have been involved in a project together with the University of Florida at Gainesville in order to in order to design a predictive model of the evolution of the viruses like SARS-CoV-2 and trying to analyze the data that are collected by public databases and detect in advance if a certain variant was going to be damaging. Actually, this kind of exercise has been performed twice. First, we started with the problem that it was clear at the beginning, that is to be able to detect if the variant was one of these three classes that is neutral, of interest, of concern, before they are basically judged as such by, for example, CDC. And actually, working on this problem have generated a number of ideas related to the issue of having a tool that is able to adapt itself. One of the challenges, for example, was that the very nature of the database changes abruptly over time. So you can see that the number of non-neutral variants became the majority along 2021. And so the nature of the database that you have to learn with moved from being unbalanced with neutral variants to be unbalanced with non-neutral variants. So we had to completely change our strategy in order to make the system adaptive. At the beginning, it was the model that we used was a classification model, but then we understood that the best way was to move to anomaly detection algorithms. And again, what we have to do was basically to design a system that is able to first understand what's an anomaly and then adapt the definition of the anomaly while the data are collected. We started with a one-class supervector machine to do this, and then we move to deep autoencoders. And what I just want to show you that we had performed a simulation that for some of the variants, we were able to perform a correct prediction of the potential dominance of the variants almost 1 year before it became dominant. So I'm now arriving to the conclusion. So my point is that now it is true that we're still waiting for the flow of AI applications coming and arriving in the clinic, but we are starting to have a consistent number of applications that are considered medical device and are based on AI models. So to this end is key to have available tools to perform online evaluation for monitoring the performance and for understanding if the model that we are using is really a good model for our institution, for our case, for our patients. And there are challenges to do this, but are urgent. We need to assess reliability and pointwise prediction. We need to have tools to deal with the other problem that I haven't touched with, that's concept and data drift detection, and the issue of deciding if we need tools that are continuously retrained and with kind of safety measures if we decide to do it, or if we have a stop, let's say, a stopping rule for continuing to use a certain model if the performance are not properly, are not well, let's say. So online principles should always accompany AI model deployment in clinical practice, and that's my main message. And with that, this is my group, and so people working with me. I had the chance to spend my entire life in biomedical informatics and AI applications, when at the very beginning, you can see there on the right, it's a paper, it's Tempo Medico, is a magazine that in 1989 published an expert system that was designed in Pavia, and it was supposed to help the diagnosis of anemia. And you see three faces, one engineer, one philosopher, and one clinician, of course, all together, because that's what is needed in order to implement AI in real practice. Thank you.
Thank you very much, Riccardo. There is a very short time for one question. One short question. There is someone. Otherwise, I have one question for you, I hope very short. You focused your presentation, your work on safe AI. And this is an issue not only for health application, but for a lot of application. And so my question is, what do you think about the importance of the relationship between the different sectors of AI? Today, in these days, we discuss about health, with all the other section, let me say, of AI working and facing the safe AI issue.
Well, in general, yes, AI is part of the safety, let's say, I would say the other way around. Medicine is one of the safety critical application of AI. That is, first of all, the first point that we have to say. To this end, it shares it may share some of the methods and tools with the other area where safety is a crucial concern. However, I always believe that medicine is so peculiar because of the interplay that we have in medicine between many different human actors that need to be taken into account. So it has special peculiarities, let's say. It has peculiarities. So it may share methods, tools, but requires its own approach in order to be able to have safe and trustworthy applications.
Thank you very much. Thank you, Riccardo.
So we move to the next talk. So I welcome Professor Gino Gerosa on the stage. So Professor Gino Gerosa is a professor of cardiac surgery at the Department of Cardiac, Thoracic, Vascular Sciences and Public Health of the University of Padua. He's also director of the Cardiac Surgery Operative Unit at the Padua Hospital, and his talk will be the impact of AI, telemedicine, and new technologies on cardiac surgery.
Thank you. Good afternoon. And first of all, I would like to thank the organizers for kindly inviting me to this meeting to discuss about the role of artificial intelligence in the field of cardiac surgery. So when I was preparing my presentation, here it is, I guess. Yeah. The first question that came to my mind was, can artificial intelligence be considered a leading actor in this field? And I thought, I doubt it. It's not the leading actor. It can be a supporting actor, but it's not the leading actor. Because the leading actor are other two, and I will show you which are the leading actor. But no, nowadays surgeons must be well aware of the role of artificial intelligence and its application through GPT, the generative retrained transformers, to fully understand what can offer to the clinical care, the decision-making, the learning process, the research and education in cardiac surgery. So we can say that GPT should be seen as a tool to support and enhance the skill of surgeons rather than a replacement for their experience and judgment. So there are several potential applications of artificial intelligence in cardiac surgery. No doubt about preoperative planning and simulation. Of course, analyzing patient data, past medical history, diagnostic reports, we can define the risk score of the patient, and we can use the AI in order to suggest appropriate surgical techniques and recommend the optimal treatment for those patients. But also can simulate different surgical scenarios, and therefore AI can act as a mentor for younger surgeon or an advisor for the senior colleague. Other potential application can be the surgical assistance and decision support. So real-time decision-making is crucial during cardiac surgery. And of course, GPT can act as a virtual surgical assistant because the surgeon can have at their fingertips the most up-to-date information in order to solve the surgical problem that can encounter. But more than that, we can use AI for intraoperative monitoring and analysis because we can process real-time patient data, and this real-time patient data can alert the surgical team during the operation for any potential complication. And also, a very important role is the recovery phase after the operation. This is extremely critical for those patients. So prehabilitation, post-operative care, and rehabilitation are of paramount importance. And I just give you an example showing one of the projects that we are actually running at our institution. The project is called Cure at Home. The lead physician is Chiara Tessari, who is sitting somewhere in this room. And we are using artificial intelligence application to improve at-home rehabilitation program in heart transplant patients and ventricular assist device recipients. So when we are talking about patients surgically treated for end-stage heart failure, of course, we have patient undergoing heart transplant, a patient implanted with an LVAD. So rehabilitation is important because we have the potential to improve functional capacity and metabolic control in order to reduce the rehospitalization for adverse events, to prevent cardiovascular risk factors, and of course, to reduce the death rate of those patients. So center-based rehabilitation is unfortunately often difficult during out-of-hospital follow-up. And then an alternative can be the so-called tele-rehabilitation. So the Cure at Home project has several steps. Step one, I think we missed the battery in this device. However, step one is a multidisciplinary evaluation at the baseline after 6 months and 12 months of follow-up. And of course, we are looking at clinical exam, we are looking at the clinical parameter, we are looking at cardiac imaging technologies, and also psychological evaluation, like the SF-36 quality of life test. And also, we're using a dedicated and patient-targeted app mobile with several sections. We are talking about moving the patient, food, psychological, informative, and data analysis with machine learning techniques for patient-targeted recommendations. And this one is one of the trend of the patient's daily parameters. That means we are assessing blood pressure, the heart rate variability, the weight of the patient, the sleeping time. And according with all this data, we can make a closer link between patient and physician, shifting the paradigm from disease to health. And so the patient have an increased engagement and self-consciousness, whereas the physician have access to earlier and rapid information. Another potential application, of course, relies on research and innovation. So the integration of GPT into clinical cardiac surgery can facilitate expensive data collection and analysis. And this, of course, can be of extreme importance during research. But also for training and education, because the AI is able to create virtual scenarios that can replicate a common or complex surgical procedure. And therefore, young trainee can be exposed to some type of surgery that will be more difficult in the real OR. Another potential application is represented by the emergency situation. No doubt that cardiac surgery is a high-stress scenario, is a sort of battlefield, the OR in cardiac surgery. And therefore, to have a quick access to information about specific emergency cardiac scenarios, surgical technique, or the latest research can aid in problem solving and potentially improve patient outcomes. Another application that are actually utilizing at the moment in Padua is the ability to support surgeons in underdeveloped countries by acting as a virtual consultant. In other words, bridging the gap between limited local resources and specialized cardiac expertise and experience. And we are currently utilizing this in order to help cardiac surgeons from Kathmandu University that would like to perform the first heart transplant in Nepal. However, it is crucial to acknowledge that GPT should always be considered a tool to augment the existing healthcare professionals rather than as a replacement for human judgment, experience, and wisdom. And also there is another issue, a caveat, I would say, that relies on money. Of course, extensive training on large data sets of cardiac surgical procedure and heart transplant cases will be required to ensure the model's accuracy and reliability, with strong impact on cost. And if you look at this pathway, if you can reach this slide, if you look at this pathway, ranging from dataset curation, iterative deployment and feedback loop, conversational interface, balance accuracy and freedom, and rest bias. Of course, we shouldn't see this like a vicious circle, but you should look at this like a virtuous cycle. This is the difference. Going back to artificial intelligence in cardiothoracic surgery, this may be applied to change surgical techniques. Let's think about remote controlled robotic surgery. This can improve the safety of intervention where clinicians are exposed to high dose of radiation and make surgery possible in subanatomical location otherwise not reachable by human hands. But again, we have some remarks. First of all, the implementation of AI raises ethical concern, including potential job displacement, but I don't think this relies on cardiac surgery. Human oversight and intervention will always be necessary to ensure patient safety and to make complex decisions that may require a better understanding of individual patient circumstances. Now I'm going to take you at the end of my presentation to the other two leading actors. So if we think about this technology, that is the transcatheter technology, usually utilized by interventional cardiologists, this is a technology idiot-proof. That means using this technology, you can perform this procedure here in Padua, in Piove di Sacco, in Mazara del Vallo, in Riyadh, wherever. But surgery is actually craftsmanship. It's craftsmanship because it's a technique rather than a technology. Technology, no doubt, is dynamic. Techniques is more static, but technique relies on human factor. So this is the other leading actor when I mentioned the fact that artificial intelligence is not the leading actor. So human factor, why human factor? Because we can think that the artificial intelligence, I think it's something like this. All of you know very well indeed this cooking device, very well known. I don't want to mention the name for publicity, but you know it very well indeed. And I believe this is artificial intelligence, whereas the chef is the human factor, and there is a terrific difference between that cooking device and a chef. So going back to the human factor, I give you another example. So let's talk about the ability to overcome the boundaries of heartwarming ischemia in donation after circulatory death. So you know that in May 11, last year, we performed the first heart transplant from a DCD donor, a dead cardiac donor, with a heart that had an asystolic time over 20 minutes. Now, let's think about the human factor, my friend from the Flintstone. So artificial intelligence was scanning the report in the literature, so I was looking at this paper, artificial intelligence stating early US experience with cardiac donation after circulatory death using normothermic regional perfusion. You see the warm ischemic time is that period from the withdrawal of life support, that is called the agonic phase, then the asystolic phase, and then the reperfusion. And they thought, and that was published, that the donor age should be well below 30 years of age. And more than that, the warm ischemic time should be well below 30 minutes. So if our artificial intelligence would scan the published report, would say, and we would put the question, can I do a DCD heart transplant? Yes, you can do that, but the donor should be younger than 30 years of age, and the functional warm ischemic time should be lower than 30 minutes. But here comes the human factor. That means the Padua case, with a functional warm ischemic time of 45 minutes. Now, it's not important, the fact that this has been done for the first time in the world in Padua. It's important that I'm presenting the data from the earliest result of an Italian prospective clinical study on heart transplantation after 20-minute no-touch period before CDC heart retrieval. And this trial is called DCD.IT trial. And we're talking about 7 centers in Italy, and according with this data, you see that the mean donor age is nearly 50 years. It's not below 30. And the other striking point is the fact that the functional warm ischemic time is not less than 30 minutes. It's even above 40 minutes with a median time of 39 minutes. So it's possible, it's feasible, and more than that, it's safe, because all those 19 patients transplanted with the DCD heart with the ischemic time above 35 minutes were successful. So this is a human factor. Forget about the one in the middle there. Look at the other 3. And those 3 are the human factor. The anesthesiologist in Treviso, Paolo Zanato, made that transplant a reality, then the surgeon, but that is not important. But more than that is the patient in the middle who trusted us. So the leading actor, if the supporting actor is the artificial intelligence, the leading actor is the human factor. But there is one more leading actor in cardiac surgery that is called courage, because which are the rules of medical innovation? Experience, creativity, and courage. And then coming at the end of my presentation, quoting Paul Torrance, when he says, it takes courage to be creative. Just as soon as you have a new idea, you are a minority of one. And the last sentence comes from Stuart Levine. Having the courage to realize new ideas is always more critical today. However, courage makes change and innovation possible. Courage is vital to challenge current thinking and to invite new possibilities. Thank you for the attention.
So thank you, Professor Gelosa, for the wonderful talk. Is there any question from the audience?
The 2 slides, of course, were for these young people, these young researchers, you know. Those slides about courage was for you. Just be courage.
Okay, I have a question. Professor Meneghes.
I want just to say thank you very much, Gino, for this talk, and you give us a very good lesson today that, of course, artificial intelligence can help us, as we have seen in all these days. He will never replace the doctors, he will never replace people like you. And indeed, this last part in which you mentioned about to be to try to be innovative. Of course, artificial intelligence cannot be innovative unless you tell them to do that. So this is what we need, people like you to train the good artificial intelligence algorithms. So thank you very much.
Thank you.
So if there is no other question, I would thank Professor Gerosa again. And I'm calling Professor Barbara Di Camillo on the stage. So Barbara Di Camillo is a professor of computer science at the Department of Information Engineering of the University of Padova. It's a pleasure to have you here. And so the title of her presentation will be Bringing AI into Clinical Practice, a deep dive into the Brain Teaser project.
So good afternoon to everybody. Thank you for this invitation. It's really an honor and a pleasure to be here. I'm going to talk about AI, of course, but through an example. The example is the Brain Teaser project. And I've chosen this example because this project was meant to bring AI into clinical practice, so to drive along all the path from data to application. And the objective of Brain Teaser is to bring artificial intelligence at home and in clinics for patients and for clinicians, of course, focusing on amyotrophic lateral sclerosis and multiple sclerosis patients. And as you can see in the slide, the idea is to provide patients with apps and sensors. The patient can be in contact with clinicians, can send data, but also questions. And the clinicians supported by AI can use the data sent by the patients, but also the clinical data that they are collecting in routine visits, and can answer to patients, can give information, can monitor them, can give suggestions, feedback, and prognosis. As we have seen in these 2 days, to bring AI into clinical practice, we need responsible AI or trustworthy AI, which means a lot of things. And among them, I'm going to focusing on data management, accuracy and reproducibility of the results, so the ability to generalize on new patients that you haven't seen before and that come to the clinic and you need to give prediction. And then to the explainability, because as we have seen again, to understand the suggestion of the algorithm is really important if you want to keep human in command approach. Otherwise, how can you decide and be responsible of the decision if you don't understand the suggestions of the AI? So I will start with data. Brain teaser data are, let's say, rich data and longitudinal data. We are collecting clinical data, longitudinal clinical data, genetic data, data collected from patients through sensors and apps, as I was saying. So they answer questionnaires, but we can also give them, provide a cognitive tests, for example, and other kind of tests. And then we are also collecting both prospective and retrospective environmental data about the quality of air. The data are anonymized before entering the Brain Teaser ecosystem. So both the retrospective and the prospective data are anonymized. They enter the system, and in the system, they can be shared within the project partner, and they can also be shared with the scientific community. They can be verified, so being, let's say, findable, accessible, interoperable, and reusable. And for example, we use them into we are using them in the last challenge, so we're using them in public challenges. So we are asking the scientific community to reproduce our results, to use different methods to try to get better results than us. And then we have meetings in which we discuss the methods that we use, the results that we obtain, because one of the pillars of science is reproducibility. So we want to assess our methods also with respect to the other researchers that, as us, are working on on clinical data. Then, the second thing I'm going to talk about is the robustness of this algorithm. We are taking all this data, we are training our model, and then the model is used to give feedback to patients and clinicians. But we need this model to be robust. And to do this, we used a framework in which the data set is split in training and test. We forget about the test that is used only to assess the performance of our method. And within the training, we again split the training set into an internal training set, which is used for learning, for AI learning, and on an internal test set, which is called out of bag, which is used to internal assessment. So in the training, we used all the good practices of AI to select the features, to select the good parameters. And at the end of this, which is important, the thing that you have to understand here is that for use of different number of features, we have an assessment of the performance on the out of bag. But we don't do this only one time. We do this many times, 100 times, for example. And each time we have a ranked feature list and the performance assessed for different number of features. And then we can average. And averaging, we have a global ranked list. We have an average performance assessment, and we can also choose the optimal number of features, which is in correspondence of the optimal average performance. Okay, now suppose we have the robust model. We have a new patient that comes into the clinic. We use the model to give a feedback, to have a feedback, a risk score. For example, 0.72. What does this number mean? So for the clinician, how can the clinician use this number? Maybe it's a probability, but if it is a probability, it must be referred to a certain time interval, right? A probability of an event, for example. So in this stage, we were discussing and, let's say, implementing a lot of solutions together with the clinicians. And what they liked most was to recalibrate the model, so to have a probability curve at different time points, which give them a probability in time, and they wanted also a comparison with the reference population to understand if this probability was high or low, also with respect to the entire population. Then they want to know why the model gives a certain prediction for that specific subject that is now in the clinic, right? So we use Shapley value, and here we have, well, it's impossible to use the pointer, but let's say in these plots, you have an idea of why a variable is protective or detrimental in terms of risk, but then you can see maybe the black circles in the central plot. Those are the data of the specific patients for which you are running the model and you are having the prediction. So you want to know for that patient how much each variable contributed to the outcome. And then we have in the last graph on the right, the distribution of each variable that was used as input for the model, but also of variables that were not used as input, but they are present in the training set, because we want to know if our subject is similar to the training set that was used for training the model, or is different, because as was said also before, if the subject is similar to the training population, then you can be, let's say, quite sure that the prediction are as good as the accuracy that was predicted. But if it's really different, it's something new that the algorithm haven't seen before, right? So it's a new experience. You cannot be sure of the prediction if the subject is really different from the subjects that you used for training the model. Then we wanted to go a little bit further, so we were asking to ourselves for which subjects our models are not good enough. And so, do you remember the out-of-bag that we were using to train a robust model? Every time a subject is in an out-of-bag sample, we can use it, he or she, to compare the results predicted by the algorithm with the truth, because if the subject is in the training set, it means we already know what has happened to the subject. So we compare the real risk with the model prediction and calculated an absolute difference of risk rank, comparing the subject with the subjects in the out-of-bag with the reality, and averaging for each subject this difference across all the time he or she is in the out-of-bag for different number of selected features. This means that you have a plot like in the bottom right, in which subjects that are difficult to be classified, we have always a high difference when you compare prediction and reality, whereas if you are able to classify a subject with a certain number of features, you will have a low absolute risk difference. So that plot was an example, but then we did it with our subjects in the project. So in these plots on the right, you see the clusters of the different subjects, and on the x-axis you have the number of features. On the y, you have the score that tells you if it's difficult or easy to classify the subject. And cluster 2 contains 166 subjects for which we are wrong, the model is wrong. So, to understand a little bit better which were the characteristics of these subjects, well, it's not shown here, but of course we compare them with the training set, and they were completely similar at the baseline with the other subjects that we classify well. So we did some archetypal analysis of the visits of the subjects. So these are the data. Each subject has a number of visits, so we put all the visits together, and in each visit, clinicians, ALS clinicians, make different questions to give a score to these 12 items. Then we can use archetypal analysis, which is an analysis which finds the archetypal visits, so the visits that somehow are representative of the status of the patients. And in this way, we identified 8 different archetypes. So you can see, for example, here on the left, archetype 1 and 2 represents an early status of the disease in which all the different items have high score. It means that there are no major impairments for the patients. And then, for example, archetype 3, the second on the top on the left, is a patient at the late stage in which all the items have a low score and there are a lot of impairments in different abilities of the patients. And then we run process mining, which is a technique to understand how you go from a status to another. So how the entire population goes from one status to another, so which is the typical pattern, and compare the subjects easy to be classified with those that are difficult to be classified. And the interesting point was that difficult subjects had a really fast or a really slow progression, but with an unexpected path, because they are good. They start from archetype 1, so no impairment or almost no impairment, and they go directly to death. Or they proceed until a status of high impairment, all the domain are impaired, but they survive longer than the other subjects. So this was completely in line with clinician experience or what they are not able to predict, which means basically that probably there are some variables that we are not measuring and that are important to understand why these subjects are really fast or really slow progressors. And now we are on trial, so we are using all these models in clinics, And we are also monitoring the performance of our algorithm. The clinicians have the progression of the disease score, so the prediction of the progression, the explainability part, but have also directly the data from the sensor. And we are comparing human versus machine prediction, but not to score, which is better, but to understand if they are if they agree or not, and when they don't agree, why. So it's more for that purpose. And also, we are measuring the acceptance and the utility of this tool in real clinical practice. So take-home message to quickly conclude. I think it's really important, since the design of these algorithms and these solutions, to keep the focus on responsible AI, which include considering data management issues, accuracy issues, but also inclusiveness and explainability of our tools. It's always important to run interdisciplinary research. This was a really interdisciplinary research. And the research is not enough, it's the last point, because now we have this project, we have 1 year in front of us, but then the project will end, and the clinicians are asking for a brain teaser too, because they want to keep using these utilities, these tools. And also the patients associations are really happy about that, but there is a regulation, there are other actors that need to come in place to go on with this work. And thank you for your attention.
So thank you, Barbara, for the wonderful talk. Is there any question? Very, very quick question from the audience. Otherwise, I have one. Okay. Anybody? Okay. So Barbara, you tried to give as many feedback and explanation of how your AI model is working with the population to both clinician and patient. But the other way around, how they reacted to all this data that actually they can see, and is that useful? How was the reaction?
Well, the data and the progression is available to the doctors, to the clinicians, not directly to the patients, of course, so there is always the mediation of the clinicians. What is really, clinicians were really happy. I think that one of the most important things was to see the data in real time, all the data of the past visits, but also the data in real time of the smartwatches, the sensors. This was really useful, but for them it was, but also for me, we were all really excited using the predictive tools, because a lot of questions came to our minds, also, okay, now I am influenced by what I see, by the predictions, so how should I react to this? And for example, we had a long discussion about that, and we decided, it was in the last slide, but I forgot to tell, we decided that an option was to anticipate a visit, for example, but never to postpone a visit. So we keep the routine, and we try to be in the safe side. From the point of view of the patients, they were really happy to have access to a lot of information, which was checked by the clinician, together with a lot of material, about daily practices, how to do this, how to do that in different languages also.
Okay. Thank you very much. Thank you so much.
Thank you.
We thank the speaker again, and we move on to the next talk.
Thank you very much. We can go ahead with the next speaker, Professor Tommaso Banzato. Thank you very much. Tommaso Banzato is Associate Professor at the Department of Animal Medicine, Production and Health of the University of Padua. And his contribution will be focused on AI in veterinary medicine, a potential bridge between human and veterinary healthcare. Please.
So thank you very much. Thank you very much to Professor Meneghesso that gave me this possibility, and I'm really happy So I know my presentation is a bit different from the others. I come from a bit different background. I'm a veterinarian. And I think that most of the physicians here might think, okay, but the title of my presentation is Building a Bridge Between Human and Veterinary Medicine. I think most of the physicians might think, okay, but why this thing might be relevant for us? Why we might be interested in this? And this is what I will try to show, or at least that's my idea. So all my presentation revolves about the idea that the pet dog is really potentially a very good model to study several human diseases. We have an increasing body of literature that is studying this concept and that is somehow trying to bring and to leverage on the pet dogs as a model of spontaneous disease. And this is also another big concept and big difference between the dogs and, let's say, for actual animal models like murine models. So we are talking about spontaneous pathology in animals that live with us. And I will try to talk a bit about the pros and the cons of the dog as an animal model. So one of the main advantages of using the dog as animal model is that we share their environment. Our pets live with us. They share our lifestyles. They share our houses. They are somehow exposed to the same pollutants. They are also exposed to our life choices. For example, dogs that live with people that actively smoke are somehow subjected to the effect and to the same carcinogenic substances contained in smoke. And for example, in this paper, we also demonstrate that the same compounds that are found in the urine of humans, of smokers, are also found in their dogs. So this is also a big similarity. We know that dogs receive an active health care. So these are very good models of disease because they somehow resemble what we do. Dogs often have chronic conditions that get treated, that get, for which they receive medications. Furthermore, the facilities that we have in veterinary medicine are becoming increasingly, let's say, technological. So, for example, CTs, MRI are more and more available in our for veterinarians, and this gives us the possibility to generate high-quality data and data that are very comparable between the two species. Another big thing about dogs is that we know that some breeds have a very high risk of developing specific diseases. This most likely comes from the artificial selection we have done on our dogs. Dogs were basically always selected for physical, let's say, traits, but in this selection, several, let's say, genetic diseases have been somehow inadvertently selected. And we know, for example, that some breeds have particularly high incidence of some genetic diseases that are usually very rare in the general population, and that also might be rare in humans. So they might become very good models to study such diseases. But the advantage is also that these dogs also live and experiment the effect of the environment on their disease. So they more, let's say, closely resemble our relationship and how we deal with such diseases. A big, let's say, the dogs are a big model to study cancer, potentially. We know that dogs have a 10-fold higher incidence of cancer compared to humans. And this is most likely also related to genetics. We know that several cancer histotypes that are fairly rare in humans have a very high incidence in dogs. And this combined with the fact that the dog have such a high incidence of cancer in general can give us the possibility to have access or to have a lot of data from this animal. This is just an example. We know that sarcomas in humans usually are uncommon. They account more or less for 1% of all the neoplasia cases. But in dogs, the incidence is fairly high. It's much higher, depending on the studies, but it's reported to be up to 25%. So we know that this, for example, for this kind of pathologies, they might represent a very good model to study such diseases. We also know that several sarcoma histotypes have similar features to the features that we see in humans. Obviously, the dog is not a perfect model. It has also a lot of limitations. We have some very obvious biological differences. One, for example, one big difference is that the link between chronic inflammation and cancer has never been demonstrated in dogs, and this is obviously something that must be taken in account. It's a big difference. There are also many others. Cancer genetics have been explored for some histotypes, but generally speaking, the knowledge about this field in veterinary medicine is very, let's say, falls a bit behind what is known in human medicine. And so this is also, let's say, a limitation that must be taken in account. There are also legal limitations. So I was obviously always referring to pet dogs, so the dogs that live with us, and the possibility to test new treatments and new procedure is limited and it's highly regulated. But we think that, for example, if we are able to move our knowledge a bit forward, we will be able to somehow deal with this limitation in a better way. Another limitation that we have as veterinarians is that we have limited access to high-end medical devices. In veterinary medicine, we have no such thing as public health care. All the care is private and all the costs are up to the owners. So very high-end medical devices, very costly devices make very poor investments for the veterinarians, so they are not really found in the veterinary hospitals. Okay, so I was trying to demonstrate how this dog could be really a good model and how we could, that we could have some information from this. But to be able to access such information, but now it's sparse, it's difficult to collect, we should, I think, act or try to do two things. Try to build integrated research facilities. These are meant to be research and care facilities where doctors and veterinarians work side by side to create a synergy between the two, both the physicians and veterinarians. There are such facilities in the US, and in some papers they are reported to be very effective, and this could be, let's say, could give a mutual interaction that could be quite beneficial on both sides. These facilities are not really enough, mainly because, as I mentioned before, veterinary hospitals are mainly private practices, and obviously they work on different, let's say they are less interested on research, but we need to create integrated research networks involving both private and public practices like university, helping a bit to, let's say, collect sample and create a network to give, to be able to get to a point where we can really compare the pathologies in the two species. Comparative medicine is a big topic. And to compare, let's say, the pathologies between two species, we need to act and to look at the pathologies at multiple levels: molecular, genetic, imaging, clinical, histopathology. So there's a lot of work to be done and a lot of different things that must be, let's say, coupled and must be connected to have really the big picture. So in our vision, this is where artificial intelligence could really make the difference. So we have heard before that multimodality learning is a very effective way of analyzing data coming in different forms, like genetics, histopathology, molecular imaging. And we think that if we will be able to create structures that will allow us to collect such and to build large-scale databases of some selected diseases, we'll be able to leverage on the ability of deep learning to see the bigger picture in the end. So just a couple of words about what we have been doing in veterinary medicine with artificial intelligence. I think that here in Padua we can be proud to say that we are one of the first, not the first, but one of the first groups to use artificial intelligence in veterinary medicine, and we are also one of the most active groups. Also, we know that artificial intelligence is quite in an earlier stage compared to what happens in human medicine. It has been mainly developed for diagnostic imaging and internal medicine. Talking about diagnostic imaging, that is my specialty. It has mainly been used for plain radiographs and, for example, the experiences with more advanced modalities are still in a very early phase. But recently, with Professor Müller, with Professor Azzori, we have been very proudly, we were being able to create some of the first models using an interspecies training. So we have been basically, we have done the pre-training of a self-supervised deep learning network on a mixed human and canine radiograph database. And finally, we did the fine-tuning on a database of canine images only that were previously used for other studies in which we have used just canine images. And we were finally able to improve the performance of our model compared to our previous experiments. So this somehow demonstrates how we can leverage between the two species. So this is a bit what we are. It's our vision and what we would like to do. Thank you very much.
Thank you. Thank you very much, Dr. Manzato. And are there some questions from the audience? No question. I have just a question or comment for you. Probably more of a comment than a question. Because I thought about the possibility to AI as one tool for the possibility of AI as a tool for One Health, and just your presentation provide me this, and I would like to ask you for your opinion about.
Yes, I would have liked to talk about a lot of different things, but I had no time. Yes, I think that obviously One Health is a big topic, and there are a lot of tools that can be used. AI is something that can really help, provided that we are able to, because AI needs a lot of data, and the creation of high-quality data is, I think, the bottleneck in this moment. So I think that that could absolutely be a very interesting topic for AI, absolutely. You're welcome.
Thank you very much. And so thank you. And we can pass through Let's begin.
Okay, so I'm calling Marco Zorzi on the floor. So Marco Zorzi is professor at the University of Padova, where he teaches artificial intelligence and cognitive psychology. So his talk will be AI in stroke, neuroimaging from brain to behavior. So we welcome the speaker.
So thank you for the invitation, and also thanks to the audience for making it almost to the end, the wonderful but the last. So now I'd like to clarify that the topic I'm going to talk about today is not so much about neuroimaging per se, but it's more about the last mile, the last kilometer, which is often the hardest also in telecommunication, if you think about it. And this is how we go from the brain to the behavior. And of course, in this particular case, I will talk about stroke patients, but it's a very general issue which also applies to a variety of other neurological and psychiatric conditions, but also to some extent, it's widely applied to healthy people, of course, always in the service of understanding better the brain, but also with relevance for clinical applications, although I would like to be clear that we are still in the sort of the early days. Now, you already heard a bit about how AI is used in imaging and neuroimaging. Even there were some talks that touched on this point. It can be used for image segmentation and classification. For example, we also heard about identifying stroke lesions or tumors. We may also use these AI techniques to identify alterations in the morphology, the function, the activity, or metabolic activity in the brain to be used as biomarkers to support diagnosis of neurological and psychiatric disorders. But as I was saying from the very beginning, what I'm focusing here today is on predicting cognitive and behavioral deficits also in the long term. For example, a year after following the stroke, following the brain insult, from the neuroimaging data. As well as, there will be less time to touch on this, on predicting response to neurorehabilitation. In other words, we'd like to also identify biomarkers of recovery of function. Now, I will also spend now just a couple of minutes on explaining a little bit what stroke does to the brain, also how it affects behavior in particular. So the stroke, in terms of the cognitive and behavioral consequences, reflects three different factors. First, the distribution of structural brain lesions, which is all but random. So this is, for example, shown here. The spatial distribution of lesions is systematic. Of course, it goes with brain vessels. It reflects how the brain structures receive blood. But also it reflects the functional organization of the brain. The brain is organized functionally in different regions, and we also know that different brain regions support different cognitive functions and different behavioral capacities. Third, the brain lesion most likely also causes disconnections between brain regions. And this is important because for example, what you see here on the bottom left is how from the lesion that you see in red, we actually do see a much more widespread disconnection between regions in the brain. I also mentioned activity, and we also know that stroke also affects spontaneous brain dynamics. And this is important because, again, spontaneous brain dynamics is not random. These fluctuations in brain activity are all but random. They reflect connectivity of structural brain networks, and they actually predict task-related activity and behavioral performance. So all of this is really important, again, for understanding the consequences of stroke. Now, how do we go from brain to behavior? So AI is used in a way that is almost the opposite from what classic neurology and neuropsychology has been done since the 19th century. Typically in neurology or neuropsychology, we would try to link a specific cognitive deficit, such as, say, aphasia, to the most frequent lesion correlate. So we say, okay, there is a lesion there which frequently is observed in this type of patients. So in AI, we try to do the opposite, the reverse inference. So we try to predict the deficit from the lesion information, and which is challenging because there's a high variability in the lesion patterns. Moreover, we are not trying here to just say this patient has one deficit, two deficit, this deficit, that deficit. We are trying to predict a continuous score, which is the performance at the test. And this is done on a heterogeneous sample of patients. So to complicate the matter, I already explained that we would need to consider structural, but also functional disconnections caused by the lesion. And we also need to consider to what extent the different types of data are readily available in clinical practice. So some type of data are acquired in routine scanning, such as structural MRI, standard structural MRI, but that's not the case for resting state functional MRI or even diffusion MRI, even less. And so we need to weight this in terms of whether these methods will be eventually useful in the clinic. Finally, the predictive ability of the AI model must be explainable. There's been a lot of discussion about this, so we must be able somehow to interrogate our model and understand what in the brain really drives the prediction. So here's a very quick summary of what we have been doing over the last 5 years or so. I will not be technical at all, just I want to convey the idea of how machine and deep learning can be used in this scenario. So first of all, as I said, we are trying to predict continuous scores from extensive behavioral and cognitive testing, not just like a yes, no, a classification, or a scale, a clinical scale. So we need to cast this as a regression problem. Second, we have lots of values to work with because the input, which is a brain volume typically, is very high dimensional. So we're going to have something like 900,000 voxels at the 2 millimeter resolution, which means that we first need to reduce dimensionality using unsupervised learning techniques such as PCA, but there are many others. This will still be a bit too much, and then we need to apply feature selection to reduce the number of predictors, and then finally we can try to predict our scores using regularization methods in regression, such as ridge regression or lasso, or even deep learning for that matter. Trying to prevent from overfitting, because that's really important, because we are working with small sample sizes. We do not have access to large samples because there might be lots of data with just images from stroke patients, but not with extensive clinical testing and with the segmentation of the actual lesion according to the gold standard, which unfortunately is still the neurologist's eye. Now, here's some results from what we can see when we also compare machine learning, so shallow learning methods and deep learning methods. The idea, of course, of deep learning is that we get rid of all of the pre-processing steps. So we do not need to do feature selection or feature extraction, and we go straight from the voxels, the 3D volume, to the prediction of behavior. And what's the result here? In general, we can fairly say that there is no significant advantage of using deep learning. And that's, again, largely due to the small sample size. The predictions are fairly accurate, both when we use shallow and deep learning methods. By the way, as already mentioned today, deep learning has a black box nature that also limits the possibility to explain the predictions. Predictions also work in the long term. So what I was showing earlier is a prediction 3 months after stroke. Here's even 1 year after stroke, and predictions still hold. But there are some caveats which I'll touch on in a few seconds. So first, I already mentioned the idea of disconnections. Here we tested to what extent having information about disconnections, either structural or functional, is important. And the answer, again, is this depends on the type of data and depends on the type of behavior that you want to predict. So there is no simple solution, there is no simple recipe, okay? Disconnections actually sometimes are very, very predictive, very helpful, for example, for predicting motor performance, so motor impairment and motor recovery, but much less for others, and some methods do not work well at all. In some cases, having access to the functional activity is important. In some other cases, is not at all important, is actually leading to worse accuracy in a prediction. And so, again, the type of data is a critical issue, but this interacts with the cognitive domain that we are trying to predict. Optimizing the pipelines is also, turns out to be important because there are many choices that are available in our toolbox in data science and in AI. We can use a lot of different techniques for feature extraction. We have different methods to carry out feature selection. We have different methods for the prediction part, for the regression. And again, this can lead to different results. So again, here I'm trying to say there is no one-size-fits-all solution. It's the more the problems are complex, the more require to really understand the problems and to explore a variety of different solutions. One of the last things I want to talk about is how we can, again, go back to deep learning. This will resonate with what some previous talks by previous colleagues have touched on. The idea that we can use techniques such as transfer learning, which was mentioned a couple of times, which is the idea that we can pre-train a model on a different type of data set. And in this case, it will be the data from healthy people. This can be particularly useful if we work with connectomes, with functional connectomes. Then we fine-tune on the patients. This can also be further combined with data augmentation, and all this goes into an unsupervised deep learning model, which is the autoencoder, convolutional autoencoder that was also mentioned in a couple of previous talks. And this can be, again, very helpful, and it can lead to large gains, but only in the very difficult situations, in the most difficult domains. So for the ones that had already quite decent accuracies with the shallow models, there is absolutely no advantage. So there's no reason really to go to deep learning and to more complicated pipelines and to less explainable AI tools. I'm really out of time, so I mentioned the idea that we can try to predict the effect of rehabilitation. That's something we are actively working on. This slide is just like a reminder for myself, but it also points to the fact that, for example, if we try to relate motor deficit to the brain, we can find a classic finding, which the disconnection of the cortical spinal tract is the main predictor of motor deficit. But if we look at the probability of recovering to the actual change due to rehabilitation, we do actually find a different white matter tract to be more important, which is actually related to the attentional function in the patient as opposed to the mere motor function. And this is the last slide before the conclusion. Much of what I was talking about can also go into the even more complex idea of developing personalized whole brain models of the patients. So each patient is represented by a personalized model based on the neuroimaging data. In this case, we use the tractography, so the structural connectome, as the basis for a full-blown whole brain model which actually simulates brain activity. And this allows to study the brain dynamics also from the point of view of statistical physics, which is interesting because it speaks to the issue of the brain being or not being in a critical state and in recovering criticality as a predictor of recovery of function. So that's really it. So I hope I was clear in explaining how AI can be an effective tool for learning brain behavior mappings, but the predictive accuracy, even for the very same patient, is modulated by the type of data, so the type of input, the target, so the cognitive domain that we are trying to predict, the choices in the machine learning pipeline, which means that we need a lot of expertise in AI, but we also need a lot of expertise in neuroscience and neuroimaging to understand the problem and to visualize the features that drive the predictions, because they have clinical relevance and they are crucial for explainability. That's something I did not really talk much about, but I can assure you that when we do that, we see that the prediction is neurologically and neuroscientifically sound. So we see what we expect to see. And I also mentioned the promise, in a sense, of deep learning when coupled with data augmentation and transfer learning for application to the small size data sets that are often a major barrier to developing these larger scale systems. The challenges and also some of the work in progress is that we can explore multimodal models. A key issue is out-of-sample validation and other type of data, and how to share data between hospitals is a critical issue. Thank you.
Thank you very much, Professor Zorzi. Unfortunately, we have gone out of time, so we have no time for questions. Thank you very much.
Thank you.
And we will go through the last but not least speaker, Dr. Alfonso Pozzan. Alfonso Pozzan currently leads the Molecular Architect Department of Evotec Verona, where he collaborates with computational chemists and medicinal chemist to drive innovation in projects focused on discovering lead molecules. The title of his presentation will be AI and Machine Learning in Drug Discovery. Please.
Okay, thank you for the introduction. And for those who doesn't know me, probably many of you, I'm currently leading, I have a pleasure to lead a group of talented young people like you guys, and many of those came from the University of Padua, from the Department of Pharmaceutical Science, where I also graduated 30 years ago and did also my PhD there, and then moved to Big Pharma, and now a partner research organization to help to discover new drugs. So the topic is how we discover new drugs that, of course, are part of the health, and this is why we are here at this Congress, right? It is also quite remarkable that 30 years ago, I was much more handsome. I look more like you than I look now, but this is how things goes with age. I would like to start with the challenge that for drug discovery. And the challenge that we have are very challenging, of course. We have about 12,000 diseases that are reported in the disease ontology database. And for those diseases, we have probably 1,000 potential pharmacological treatments, some related to orphan disease, some related to more common disease. Just to mention that last year the WHO released the list of essential drugs, and this are just 600. So we have an order of magnitude of less pharmacological treatment than what is supposed to be the disease. And this is just treatment, are not cure. The other challenge, of course, is that the potential number of molecules that could be synthesized are a huge amount. 10 at the 60 is pretty much the size of the universe. And amongst those, there are probably a lower number of potential molecules that could become drugs. And those are probably at the 10th of the 9th. So it is quite challenging to find those amongst the huge universe of potential molecules. And those molecules at the 10th of the 9th, they won't be all active. So it's like finding a needle in the haystack, but we don't know even where is the haystack, right? Also, there is a huge amount of information. Elsevier, which is one of the major providers of scientific paper, has 1.8 billion articles, and only in 2022, there were 2.7 million articles published, yes. So a lot of information, which is difficult to find the right one, and sometimes there are errors, so it's very challenging also to exploit information. Another challenge is that we are not only cure or try to find cure using drugs, small molecule, the one that you buy at the pharmacy or the one that gets mostly prescribed by physician. But now there are also new, there are antibodies, there are proteins and peptides, there is cell therapy and gene therapy, nucleic acid, oncovirus, and also protein degraders, the PROTAC. So this makes things even more complicated. And then there is the cost. The cost is 2.2, on average is 2.2 billion to develop drugs, and it takes 10 and 15 years on average. And for example, gene therapy, the cost for a single treatment is now 3.5 million for a single treatment from one patient. Of course, no wonder why now there is a big expectation that AI will solve some of those challenges. Okay, this is just how machine learning and artificial intelligence is applied throughout the value chain of the drug discovery process. And we know that the drug discovery process is more or less divided in 3 phases. The first one is the target identification and validation. In that case, we are not discovering new drug, we are just working on the new target. We have seen many lectures where they were dealing with the omics data, or data also from clinics. And of course, there is a lot of AI machine learning in that area. If we go on the right-hand side, there is a preclinical data. Also in that case, there is not a lot of drug discovery because the potential drug may be discovered. We are preparing the drug to enter the clinical studies. Further on the right, there will be the clinical studies, and then, of course, there is no more drug discovery because the drug has been discovered. The expectation is that it doesn't fail in the clinical study, and then we can be delivered into the market. So the core of drug discovery is the central part. In that part, really, we fail a lot, but we try a lot of potential drugs. And this is where my department, the molecular architect, is working. The green area are all activities where machine learning is applied by the team that I'm lead. And this is very similar to what happens in any pharma company or biopharma company where those activities get carried out. At the bottom, there are data. All machine models are, of course, trained by data. This is very important that the quality of data is very high. Okay, so how AI and machine learning evolved during the time as part of drug discovery. This is a very simple attempt to identify the importance of AI and machine learning by monitoring the number of publications that were containing AI and machine learning associated with drug discovery. And you can see that up to 2014-15, the growth was quite significant, but not like the one that we see now, starting from 2016-17. I started working in 1990, and of course I was using a lot of AI and physical methods to support drug discovery, but there was not a lot of AI at that time. At a certain point, it was about 2016, everybody was talking about artificial intelligence. And this is even after that, for example, IBM Deep Blue beat Garry Kasparov in a chess game, or even after AlphaGo beat humans playing Jeopardy on TV show. I don't know whether you have realized that before 2016, when you were translating from English to Italian, you were having a very funny translation, and sometimes you were laughing. After 2016, toward 2018, the translation became very accurate. This is where also things changed dramatically as part of AI applied to drug discovery. I'm not sure whether this is happening also on your discipline, but there are at least two key papers that sign a little bit this transition in the natural language process are the paper from concerning the use of transformer and BERT by Google, and those were really the paper that signed the big improvements of artificial intelligence, and probably this propagate also on the other discipline, including drug discovery. Okay, this slide is just to highlight that as part of the drug discovery, there is a variety of algorithms that can be used and that belongs to machine learning. I just collected the one that we are using in our team, and there is a little bit of the perception that the one on the left-hand side is more perceived to be traditional, while only the one on the right-hand side are perceived to be artificial intelligence. But in reality, they are all part of artificial intelligence. So the key take-home message here is that, of course, AI and machine learning has been part of the drug discovery from many years. And I have reported here two publications. One is data in 1964 and one 1976, and these for me are one of the initial paper concerning machine learning. So what happened towards 2016 that really wasn't available before? So when I was working throughout my career, I was really you see this this person in front of the computer where it's written a feature extraction. This is me 20 years ago, 30 years ago. I was really taking information from the molecules that are represented on the left-hand side. You see there is caffeine, then there is this funny aromatic molecule, and then there is a sort of protein. This is me trying to extract feature from the molecule and then build a model and then try to predict the activity of those molecules or classify those molecules. This is something that I was doing until 2016, and this is and I'm still doing. But as I mentioned, in 2016, Transformer has really changed the way in which we process natural language. And the same happened also in chemistry. In 2017, a key paper from Gomez, Bombarelli, basically they did the same of language transformer with molecules. So basically, they build a dictionary that can transform molecule into molecules. And by building this dictionary, this translator, they were able to model this dictionary and so generate a molecule that could optimize some of the properties. Basically, it's exactly the same as building a translator from English to Italian. In this case, they were able to build a translator between a representation of a molecule to another representation of the molecule. And this is really something that I think is a key moment in drug discovery, because allows to do what is represented in the bottom part of the graph. Now, there is no need to have the person in front of a computer to extract feature from the molecule, but we can use a representation of a molecule and directly build a model and classify the model. So the advent of transformer and generative model, of course, allow to have generative model. So now what we are doing in drug discovery is to train those generative models using a big database of data. For example, the Campbell database is a database containing millions of molecules that has been generated as part of a drug discovery process. Once those models has been trained, we can then generate molecule and then score against physics-based model trained on the experimental data, and then the concept is that we can improve and forward back this information to the generative approach, and this is usually generate molecules that fit the experimental model. Okay, it doesn't move forward. Sorry. So, is it that easy? Are there any limitations? Yes, there are big limitations, and this is why we are not seeing the same impact that generative approaches or transformer has having in the natural language process or in the image processing. One of the reasons is that at the moment, those artificial intelligence and machine learning is applied to a representation of a molecule. Here you see ibuprofen, which is represented like a graph, or it can represent as a smile, which is the text below the graph. But this is far from being a real molecule. We knew that, for example, if we represent the molecule with the volume or with this conformation, so the dynamics, in 3 dimensions, this is a better representation of a molecule. But still, this is not the reality. And this is why artificial intelligence applied to drug discovery is not one of the reasons, because it's not having the same impact as in the natural language process or image processing, because the representation of a molecule is far from the reality. The second problem, of course, is the experimental data. We are generating experimental data throughout all the drug discovery process, and we are generating data from isolated target, from cellular data, from tissue. It's difficult to generate data from organ, and of course, it's difficult to generate the data from the human being or reflect on the population. We can have clinical data once the drug reaches the market, but of course, we can test our drugs directly on the human being. So there is really a long really a big difference between the data generated on isolated target and what happened once the drug reached the population. The combination of these two aspects, of course, is still one of the limitations for which the artificial intelligence is not having exactly the same impact like in other areas. This is, before we get into conclusion, this is my last slide. There is another area, of course, where artificial intelligence can have an impact. This is application of a GPT-4 by OpenAI. Last year, as part of the experiment that they are doing on OpenAI, they tried to build a team, a red team, where they implement a sort of drug discovery process as a textual input. So you can see this is, the text say, propose a potential inhibitor of a target X, and blah, blah, blah. This was actually implemented. So we call now this sort of augmenting the natural language process or augmenting the GPT-4. But of course, behind the scene, there are tools that perform complex tasks. So I think that's one potential, one great potential for artificial intelligence in drug discovery is really the use of GPT to interact with the complex system, even if this is not really an intelligent system. Behind the scene, there are all the tools that perform the complicated task. So, and we can go to the conclusion. Artificial intelligence indeed has had a change and an impact, in particular post-2016. This is mainly due to the advent of autoencoder and transformer. There are, of course, limitations that I mentioned, so it's not having the same impact like image recognition or natural language processing. However, there is definitely a lot of input now and a lot of potential to use these approaches. And really, now I start to see generative approaches that generate molecules that could be suggested by human beings. And then, of course, is about integrating this system with the human in the loop. So there is still plenty of work to do for us, and there won't be any artificial intelligence system yet that will design things for us. And that's the end of my presentation. And really thanks for being still here.
Unfortunately, also in your case, we have gone out of time. So we have no time for questions. Thank you very much.
Okay. So we thank the speaker again. So I think we can close the session.
Now.
Thank you very much for being here. Thank you for staying here till the end of the session. It was pretty long today, sorry for that. And I believe we are ending these 2 days of conference giving some messages to you, right? So first of all, most of you are students to the medicine or maybe other departments. So let's say the future, if we wanted to be on the line of the development, please consider that you have to at least know artificial intelligence. This requires the collaboration between several disciplines. This will never replace the doctors, but will support the doctors, as we have seen before also by the presentation by Gino Gerosa. And you, in the future, remember, take to know what artificial intelligence can do, and with your brain, with your intelligence, you have to understand where and when you have to use it, and when you have to rely on your brain, on your knowledge, on your capability.
Okay, this is is a complete, okay. I think that, just to add a few words, I think that for sure artificial intelligence represents a paradigmatic shift, a true revolution, but at least in technology, I hope also in the science, and, but we have to better define the borders, because now I think that the borders are quite foggy at present, and we have to better define what will be this, what is this revolution, and if this revolution will be followed by very, very important good outcomes. This is, I think, one of the most important messages that I would like to give you. And another important message is that I would thank you a lot, and also the speakers, because I'm really full of doubts, and that I will bring home these doubts. But I think that to be full of doubts is the true position for a scientist. Exactly. And this is my final message, be full of doubts. This is really a full position for a student, for a scientist, also for professors. Okay.
Thank you very much. Thank you, goodbye.